Okay, there are only about 150 of you. I guess it's very unfortunate, but I think that's something to do with imparters, but I'm really sorry about it. You're able to hear me nice and clear, right?
Yes, sir. It's clear.
Yes, sir.
Okay, that's good. Give it a minute. We can do a 25-minute class. I'm just waiting to see if about 200 people join. And you got an invite also, a separate email, or you're just going to the Teams and joining in?
So we just went to the team and joined it.
I see.
So we also got the
We have
invite.
also received an email as well.
Because I tried to send an email. Okay, good. That's good. Okay, there are almost 200 of you. That's only less than one. That's just less than 1/3 of the class, but I have to go ahead. Okay, let me see
So,
what I can do.
so there's an issue, sir. Sir, only a few people can join MS Teams. There's a limit.
Yeah, I think 250 or 300 is the limit, right?
Yes, sir. 300. 300.
Okay. I think
So
I
maybe
think
by next class,
huh?
Impartus is fixed.
Say that
Maybe by
again.
next class, Impartus is fixed.
Can you speak up a little bit louder?
I'm saying, sir, by the next class, maybe Impartus is fixed.
Imparters may be fixed by the next class. That is fine. Okay, fine. But I'm thinking of doing a 25-minute class today. It'll be recorded. I think everybody else can access it. And there aren't 250 also. Okay, so I think, what do you guys want, to go ahead or redo the class on a Saturday?
So I think we should go ahead. So you can record and put it on Impartus on the flipped lecture section.
Okay, let's see. Let's see what I can do. But okay, let me just go ahead. I think there isn't much we can do. I'll just go ahead with this class and then we'll see what we can do. Okay, I'm going to share the slides and then get started. Can you see the slides, guys?
Yes, sir.
Okay.
Yes, sir.
Very unfortunate, but let's see. Okay, it's saying people are waiting in the lobby. Okay, I'm admitting everybody possible until 250. Okay, my name is Vivek Perumal. I'm going to be your instructor for the next 12 to 13 lectures. I'll be doing all of them. Particularly, I'll be doing everything to do with immunology and microbiology, and then I'll at least introduce you to something called epigenetics. This is what I'm going to do for this course. My name, email address, and contact number are given here. You may contact me at like any time, like of your choice. You can email me also. You can also call me. Okay, if I'm busy, I'll let you know if you call me, but you can call me if you don't get any of this. Okay, so today we're going to start off with what is immunity. Like, what is immunity? It's the ability of the body to fight pathogens, but it's not restricted to pathogen. Immunity involves the ability of the body to fight cancer cells, anything foreign material, not just germs, but it can also fight like pollutants and other things. That is immunity, and that's what I have here. There isn't much in that slide. Okay, now a little bit of history always helps. You can go back and you can understand this better. So about 120 years ago, two scientists actually tried to show that serum from vaccinated individuals could actually protect animals against an organism called diphtheria. You all of you have heard about diphtheria, right? So that is, they showed that the serum contains some components that are related to immunity. Today we understand that's going to be antibodies. At that time they didn't, but they showed that. And then another scientist, a Russian scientist, Mechnikov, what he showed was that he showed that the cells, they also contain important components of immunity. In fact, he is the one who discovered something called macrophages, which I'll talk about today. Okay, and I'll tell you what macrophages are, also what they do, and everything I'll tell you about macrophages. Now, what I want you to understand is that not just the liquid part of the blood, which is serum, but also the cells have immunity. But then about 50 years after that, they understood there are some cells called white blood cells, and they are key players in immunity. So the take-home message from this slide is that both the serum, which is a liquid part, and the cells contribute to immunity. Now, immunology is an important aspect of biology. You can understand there are almost 20 Nobel Prizes that have gone to the field of immunology in the last 100 years. Now, generally, as a school student, if you think about immunity, we always think about blood. But blood is not all about immunity. The immune system is all over the body. That includes your skin, which protects entry of microbes. Your mucous membrane have some additional barriers. And then most of the tissues, including brain, lungs, liver, all of them have a collection of immune cells which help you fight infection. Now, just like we have two arms in our body, the immune system has two arms. One is called the innate immune response, or the non-specific arm of the immune system, and that's the first line of defense. And then if that is breached, the pathogen encounters the second line of defense, that's on the right side. That's called adaptive immune response. That's very specific against the pathogen, and that's the second line of defense. And of course, they talk with each other. Okay, the two arms of the immune systems, they talk with each other. Now, let's say a microbe invades our body. What are the barriers it encounters? It sequentially encounters a variety of barriers, starting from your skin, which is a physical barrier, and then there are other secretions and mucous membranes. Mucous membranes, for those of you who are not familiar, is nothing but the moist membranes that are inside the lining of the throat, mouth, etc. So I'll tell you a little bit more, you'll understand. So they also contribute the first line of external defenses, which are non-specific. And then there is another line of internal defenses. If the skin is breached, there are cells just below the skin that will eat up the bacteria or other germs that enter, and there are antimicrobial peptides and a few things like that. That is the second line of internal defenses. If this is also breached, then the pathogen encounters what is referred to as acquired immunity, or very specific immunity against a particular microbe. So this is the order of, these are the three layers of defenses. Two of them are contributed by innate immune responses. There is external layer, there is an internal layer. They are non-specific, they are very fast. If these are breached, the pathogen encounters the third layer, which is very specific, and that is called the acquired immunity. You will understand this very, very well as we go on in this class. Just to give you an intro to what is innate immunity, how does it compare with adaptive immunity, I have an analogy here. I'll give you an example. Imagine that the human body is the IITD campus. Okay, and then we'll first talk about the innate immune responses. Imagine the compound wall with the barbed wire fence is the layer of innate immune response around the human body or IITD campus. Now, if an intruder tries to jump into the campus, the barbed wire fence and the compound wall will try to stop it. And there is no time lag, right? Because it will stop anything in real time. Anybody who's trying to get in, it will stop it in real time. It'll try to harm the guy, the barbed wire fence. It's not specific to anybody. If you try to jump or somebody else tries to jump also, they will stop. There is no specificity to it. And they don't remember who tried to cross the wall. So that is pretty much exactly what innate immune system does. For example, the layer, your skin, stops all microbes from entering. It does not know who's trying to enter, does not remember who's trying to enter, and it stops it in real time. Now, imagine an intruder somehow jumped into the campus. Somehow he managed to get into the campus, and then the second line of immunity kicks in. And who's that going to be? That's going to be security personnel, for example. And that will be equivalent to second line of defense. the security person will not be able to pin down this guy, the intruder, in real time, right? There will be some lag, right? Somebody will tell them, I saw somebody jump, and they will try to search for him, and then they'll take a little bit of time to pin him down. So there is going to be a lag for the adaptive immune response, because the body has to identify something specifically and mount a strong immune response only against that. Just like the security personnel will try to identify one single intruder, our body will try to identify one single pathogen. So there is a lag there, and it's very specific. They're not going to track down everybody who's walking. They're only going to track down somebody who's going to be very specifically. They're going to look for somebody and then pin them down. Just like that, this is very specific to a pathogen, adaptive immune response. And once they find the intruder and pin him down or get rid of him, they remember, right? The security personnel will have a file with his photo and put his photo up so that other that when they encounter the same guy again, they remember and they can act very fast. They can stop him at the gate. Just like that, our adaptive immune response, once we have a pathogen and it mounts a response, although it takes time, our adaptive immune response remembers the pathogen. And next time over, when we see the same pathogen, we have a memory and we can act very fast and stop the pathogen before it can cause more damage. So that is the adaptive immune response. So this analogy perhaps can help you remember the distinction between innate and adaptive immune responses. Now, there are a variety of mechanical factors that contribute to the innate arm of the immune response, the non-specific arm. For example, the skin. Our skin is a barrier throughout the body. It stops all microbes from entering the body. Then we have something called the mucociliary escalator. Now, what is this? It's a fancy name, but it's a very simple thing. Let's say this is a tube. That's the trachea or your windpipe. If you look at it, or your respiratory tract, if you're looking at it, if you cut it open and look at it from the inside, this is what it'll look at. This cartoon in the middle, this is what it'll look like. So there is a gel-like substance that's called mucus that will trap all the microbes. And then you have hair-like structures. That's what you see in this cartoon, that are called cilia, and they always flap upwards towards your throat. Okay, why is that? Because the mucus traps the microbe, the cilia push it up. What happens if it comes to the throat? That you can have an irritation, you might cough it out, or you may swallow it, and the acid in your stomach will take care of it. So this is a way that the respiratory tract keeps out pathogens. So that is called the mucociliary escalator. The mucus and the hair-like structure cilia contribute to the mucociliary escalator. And then there's the flushing action of tears, urine, saliva, etc. All these flush out microbes. If there are going to be growing in any of these parts, they're going to flush it out. And then we have chemical factors. Like I told you, the stomach has acid. All of you know this from school, and it has hydrochloric acid, and the pH is somewhere between 1 to 2. It's highly acidic, and most microbes do not resist it. A few do, but most don't. So, and then we have antimicrobial peptides, which are like short proteins that can target bacteria and kill them. And we have in our saliva and tears, we have something called lysozymes, which will also kill a variety of bacteria and pathogens. So we have chemical factors throughout, like in the skin, in the stomach, in the tears, in saliva. We have chemical factors that will take care of microbes. Now, if you look at the biological factors, what are the biological factors? We have a variety of normal flora, which are microbes that are part of our body. If you look at the number of cells in our body, that's roughly about 1 to 10 trillion. If you look at the number of microbes in our body, they are almost 10 times more than the number of cells in our body. So we call it second genome. How do these normal flora, which are harmless bacteria that are part of our body, how do they contribute to defense? That's because they pre-colonize and they take up the space and the nutrients, so pathogens cannot easily come and stick there and grow or colonize other parts. They prevent by preoccupying the space, competing for nutrients. So that's the way they help contribute to as part of the immune system. That's normal flora. So in terms of not the innate arm of the immune response, which is a non-specific arm like the compound wall of IIT Delhi, if you look at it, the second layer that I showed you in the cartoon, the outer layer, which are external defenses, you looked at it already. Then there's an internal layer of defenses in the non-specific arm. What is that? That primarily comes from your blood. If you take a little bit of your blood and put it on the table and look at it after 2 hours, it will look like this. So you'll have a liquid called plasma or serum, and then you'll have RBCs here, almost equal, although they're slightly more liquid, they're almost equal. Okay, in between them, you have a small layer called buffy coat. It's just a white thin strip. And this strip contains the WBCs, or what all of you know as white blood cells. Now this, I told you that the plasma is also important. The liquid component is important. 2 scientists got a Nobel Prize for discovering that. That's important for immunity, and that contains 3 important components: coagulation proteins, which are important for clotting of your blood. Then there's something called complement proteins, spelled with an E. I'll tell you what these are. Don't worry about it. And there's a bunch of molecules called cytokines, which is one of the important ways that immune cells can alert or talk to the rest of the body and other immune cells, is through these secreted substances called cytokines. So 3 C's here. Don't have to worry about it now because we'll look at each one of them in slightly more detail. So at that time, you'll understand this. But that's the liquid component. This part, RBCs, all of you know what RBCs are, or red blood cells are. But then we're going to zoom into the WBCs and look at it. Now, all of you know when you get sick, you get a WBC count and you get a differential WBC count. When your doctor orders that, this is what the lab does. They identify how many number of WBCs, what type of WBCs you have. So in our blood, there are 5 different types of WBCs. That's what I have in the slide. They're called, so you can look at it. They're basically these 5 types. And if you can notice, these 2 types here, lymphocytes and neutrophils at the bottom left panel, so they contribute to almost 90% of all the WBCs that are circulating in your blood. The other 3 contribute only 10%, approximately 10%. Now, these 2 are the main, they are the major contributors to the WBC numbers. If you look at it, I'll tell you a little bit more about these cells. Okay, so what about WBCs? So WBCs, there are 5 types, like I've shown you in this cartoon. I'm going to zoom into lymphocytes. Lymphocytes, you see that they don't have these dots. Monocytes also, they don't have these dots or granules inside them. So these 2, they look slightly different from the rest of them. So these 2 are called WBCs without granules, or agranulocytes. You don't have to remember this. But this is the lymphocyte, an important player in the immune response, and they are divided into 3 types: B lymphocytes, T lymphocytes, and the natural killer cells. You don't have to memorize it now. We'll come back to it some other time, but these are the 3 types. Then monocytes, they're present in small numbers in blood, but they go to different parts of the tissue. I told you blood is not the only component where you have immune response. You have immune cells throughout your body, and some of these cells from the blood migrate to tissues and they become what we call as macrophages. Okay, that's here. Now, there's also one more thing. Okay, so don't worry about anything else in this cartoon. There are 5 types of WBCs. Lymphocytes and neutrophils contribute to 90% of all WBCs, and there are 3 types of lymphocytes, and monocytes can migrate to tissue and become what we call as macrophages. So these names may sound a little bit confusing at this point, but don't worry about it. You're going to remember them. We're going to make it easy for you. Don't worry about it now. So if you have 100 WBCs, almost 60% of them are going to be the cell type called neutrophils. And what do? They are phagocytes. Phago means to eat, cytes means cells. So these are cells that eat up cells. I'll tell you what they eat. So these are cells that eat up, they find and eat microbes. That is the function of the cell called neutrophil right here. That is what they do. They find and eat microbes, and they are the most abundant WBCs in your blood. Now what I'm going to show you in the next minute or so is, let's see if I can play this video. Can I play this video? Give me a second. Okay.
These human neutrophils take a
So we can't hear the audio.
You can't hear the audio. Okay. So what I'm going to, you just look at the video. I'm going to give you the audio. Okay. Okay. Don't worry about it. We'll finish in 5 minutes. We'll finish it. I'm going to play the video again, and I'm going to give you the audio. So these are neutrophils, and you can see how they're actually chasing. I'm so sorry. I can't speak with that voice. That is so loud. Okay, so these are neutrophils that you see on the screen where I'm pointing out, and this is a pipette. This is under a microscope. Okay, the pipette, they're just releasing small amounts of chemicals that bacteria release, and see how the neutrophil reacts. Okay, that's where we show. You can see it. Please notice how it's shifting. When they move the pipette with the chemicals, it's going towards that pipette. Can you see that?
Yes, sir.
Yes, sir.
Okay, so that's neutrophils, and they just track chemicals secreted by bacteria and then move towards that. That is their function, because these are the WBCs which can find and eat microbes, phagocytes, right? Find and eat microbes. That's what they do. Now, this is a microbe here in the cartoon. I'm just going to go back to the pointer mode. I know we have only 5 minutes. We'll finish in time. Okay, so I'll do only about half the lecture, but a little bit more than half perhaps, but we'll stop at 9 o'clock. So this is the microbe. This is the big one, is your neutrophil that's going to chase the microbe, find it, and then it engulfs the microbe, just extends some parts of its body and engulfs the microbe like this. That's what is happening here. And then it releases some chemicals called lysosomes, and that will digest this bacteria, and unwanted parts are just thrown out. That is what neutrophils do, and they have these granules inside them, and these granules have substances that can degrade microbes. And they have, what is that substance in the granule that can degrade microbe? It's called lysozyme, which is also present in tears, which is also present in saliva. And this lysozyme can actually degrade the bacterial cell wall. That's the function of these granules. It digests the bacteria inside the cells. So after it eats, there is digestion, which is normally what happens inside our body as well. Now, apart from eating and digesting microbes, the neutrophils have two additional functions. Now, what are they? If you look at it, after digesting it, the microbes, the neutrophils do something very important for the body. They hold out pieces of the microbe on their cell surface. Now, you can see that the circled part, the red circle, after digesting the microbe, they hold out some pieces of the microbe, proteins especially, on the cell surface. And what is the role? They call out to immune cells, other immune cells, and say, hey, a microbe is around, I need help. That's what they tell other immune cells. And once that happens, other immune cells can sense there is a microbe attack and they come to help. That's one function. The other function, there are only two of these. One is to hold out the microbe and call for help. The other function is to trigger something called inflammatory response or inflammation. In common man's term, let's say you get a cut, you get infected, you see a swelling, right? That swelling is what is called as an inflammatory response. Whenever you get a wound, you don't take care of it properly, you get swelling, and that is what is called as inflammatory response. At this point, if you remember that is inflammation, it's good enough. So neutrophils do three things. One, they find microbes, eat it, digest it. That's one thing. Some parts, digested parts, they hold out and call for help. Second thing, that is called antigen presentation, or they present proteins of the microbe to other immune cells and attract them. That's the second one. The third thing is they trigger something called inflammation. Right now, you just remember inflammation is equivalent to swelling. Now I have another video. So this, you can also look at it later. Let me see what I can do. You may not get the audio, but this video is good enough. Oh my God. This video is good enough. I think after this, we can just close for today's class after this video. This video shows how a macrophage can pick up bacteria. I hope you could all see the video and you've understood something about what we spoke about. Oh, that's good. I can see 335 people. So I think 500 may be the limit on Teams, but we have to close for the day. We'll do the rest of the lecture tomorrow. I'm just hoping you understood this, and sorry about the confusion. I think that's not on me, that's on Imparters. I had 2 setups ready and 3 internet connections, but sorry about that. Okay, guys, if you have any questions, yeah, go ahead and ask the question.
Our next class will be on Imparters or MS Team.
I'm going to contact the technical support team in the educational office, and then I'll send it to you all by email. You'll get either a Teams invite or you'll get an Imparters invite, depending on whether or not that's going to be fixed. Okay.
Thank you, sir.
Okay, guys. Bye.
Bye, sir.
Thank you, sir.
Bye and thank you. I'll take questions on this class next in the next class. Okay, somebody had a question. Okay, go ahead. One of you have a question, others can leave, no problem. If you want to stay, you can stay. Somebody had a question? You raised your hand? Okay, no worries. We'll do it next class.
Thank you, sir.
Yeah. Okay, bye-bye.
Thank you, sir. Thank you, sir.